Hello. So I want to talk to you today a little bit about what exactly is bioinformatics. And I want to talk to you a little bit about proteins and the role that bioinformatics plays, or the role that the databases play in bioinformatics of proteins. So, bioinformatics, in short, is just the extraction of information from the massive volumes of sequence data and biological data in general that we've accumulated over the last 30 or 40 years. It, I say the last 30 or 40 years because the amount of data that has been accumulated, even in the last 20 years, pales the 20 years before that. And the same thing with that 20 years pales the 20 years before that. Our ability to compile data, biological data, is increasing at an exponential rate. And it's just amazing how much data we have and how little of the data that we've collected has actually been properly processed. So, in essence, a bioinformaticist takes that data and tries to find some meaningful information out of it that answers a question that we have, a biological question. So, a bioinformaticist is essentially a data scientist that's answering questions in the biological realm. And in order to do that, the bioinformaticist needs to have a background in computer programming. Uh, Python's probably the easiest to learn and the easiest to apply. Therefore, it's also the most widely used. But there's also R, and then there's C Sharp, uh, VB.net. I mean, there's several of them that can be used to apply bioinformatics. Most of your initial inquiries are going to be done in either Python or R, though. So, in order to get the information that you need out of that, you need also a background in statistics, mathematics, data science, uh, as well as the programming aspect, and you need the biological background. It's hard to know what you should really be looking for if you don't have the biological background. So that's essentially what a bioinformaticist is. Now, that term has been around for probably almost 50 years. It wasn't originally applied in the same manner that we apply it today, but really, you didn't really hear a whole lot about bioinformatics until really the early 2000s. So I can remember back in the very early 2000s, in the late 90s, whenever the Human Genome Project and various other genome projects were underway. And those of us that were using scripting languages and using those to answer questions, biological questions, we didn't really consider ourselves bioinformaticists at the time. We were just answering questions that we had, and that was the best way to get at those answers, was to use our programming skills to get at those answers. We knew that there was too much data there really to manually go through, and so we applied our programming skills and learned our programming skills as well uh, as a result of that need. So later on, in probably the 2010s and the teens, the term bioinformatics and bioinformatician became more used. So the proper term is bioinformatician, not bioinformaticist. Although I've seen both used, but the proper one is bioinformatician. Uh, and one of the major aspects of being a bioinformatician is the analysis of sequence data. So I already mentioned that the genome projects. Well, as a result of the genome projects, we have the ability now to predict proteins without ever knowing that the protein is expressed in an organism. So we have predicted proteins. We also have proteins that we get from mRNA. We know that the protein sequence, because we have the mRNA, we get them from a cDNA library, and we know that it's expressed at least to the mRNA level. And then there's a smaller subset of proteins that were sequenced from, the actual protein isolates. Uh, so there's databases out there that house that data and classify that data. So one of the major players in the classification of protein data is the Structural Classification of Proteins database, or the SCOPE database. And this database is basically a manually curated database. So as you can see here, I have the SCOPE database opened here, and I searched for the word globin, and it brought up the globin-like superfamily, as well as the family of the globins. And the family of the globins has 200 different domains in it. So the domain is the basic functional unit of a protein. So whenever we're looking for associations between proteins, a lot of times we'll look at just the domain, not the entire protein. We just look at a single domain. For example, with alpha actinins, we might look at the spectrin-like domain or the actin binding domain. But less times will you actually look at the entire protein and try to compare the entire protein. And the reason for this is that those domains sometimes get reshuffled, sometimes they get duplicated. There's all kinds of factors that affect the pressures, the selective pressures on different parts of a protein. So if we want to look at the evolution of just a domain, then we pull out just the domain. And the structural classification of proteins database does just that, and it does it manually. So why would we need to do it manually if we can look at the actual sequence? We can align the sequences, so on and so forth. The reason for this is because a lot of these domains maintain their structure, uh, but they don't maintain their sequence quite as well. So they diverge to the point to where they have less than 30% amino acid identity, and when they do, our ability to align them is diminished. And in fact, as you get further and further into the sub 30% identity zone, the twilight zone is what it's called, uh, chance is almost equally likely to produce the alignments that we get as if the proteins actually evolved from a common ancestor. So there's a lot of ambiguity there. So for that reason, sometimes we need to look at the actual structure of the proteins to see how conserved the structure is, rather than to look at the sequence to see how conserved it is. Well, this a database that looks at the structures manually like this provides a great benchmark for people that are trying to come up with algorithms to actually use either the structural data or the sequence data to come up with the same classifications. Because if a human is reasoning through it, they're looking at the structural data, they're looking at some of the other data, maybe even functional data, biochemical data, so on and so forth, to come up with the classifications, then we can consider those to be pretty ironclad classifications. Uh, Normally more so than at least the first generation of algorithms to classify those same domains. So, for example, this globins family here with 200 entries could be used for a machine learning classifier to classify those 200 domains as being globins whenever there's a background of, let's say, 60,000 other uh, domains. I think this database, I can't remember the exact size of it, but I think it's about 64,000 uh, domains, something like that. So that's the reason this manually curated scope database is very important. Now, there is another database called the CATH database. And the CATH database essentially does the same thing. It classifies protein domains into different superfamilies in this case. So homologous superfamilies, that's what the final H in the CATH uh, part of name of the database is. It stands for homologous superfamily. So that's the most focused classification in the CATH database, but it tries to do this algorithmically. So uh, in this case, I have the same structure, 2DN3, pulled up here in the CATH database uh, that was the first structure or the first domain in the scope database. So a lot of the same domains are in both databases. However, if you look at the globins, it's a globin-like superfamily in CATH. There's many more members. So it's much more efficient to uh, algorithmically classify uh, different domains than it is to do it manually. So if you look at the superfamily, the globin superfamily in CATH, we find that there's actually 2,891 members of that superfamily. So the superfamily in CATH is the most focused classification in CATH, and the family in scope is the most focused classification in scope. So essentially, they are, especially with the globins, equivalent of each other. So that's a little bit confusing. The scope family is equivalent of the CATH superfamily. Okay, so I've already explained how those can be used to work with classifiers, AI, also machine learning classifiers, the same thing. It's a type of AI. Uh, and if you design a classifier using the scope data, then you can see how well it uh, generalizes to the CATH data. And just because it doesn't generalize well, if it doesn't, does not necessarily mean that you have a bad classifier. It could mean that the parameters for classification in CATH vary from the parameters in scope. So again, the scope is what I consider to be the more ironclad option, whereas the CATH is a good option, uh, but it's not as probably as stringent as the scope database. Now then, so those are two databases to where we can look at classification. Okay, another database is the Protein Data Bank and PDB. And what this has in it is the actual structural data. So every domain in the scope database is going to have a PDB identifier. Okay, and it's going to link to the protein data bank. And that's going to actually have the structural data. So if you wanted to start comparing proteins based on their actual structure, uh, there are some options out there. So, for example, FoldSeq is a uh, algorithm that compares structures based on their actual structure. Or you could use root mean square deviation, RMD, RMSD. Uh, and that's a little bit of an older form of comparing structures. And you can come up with a distance between two structures, how similar, how different they are. So if you want to use those type of data in your analysis, then you can get those structures right here. So when you come to a structure in the protein data bank, uh, if you want just the structure and you want to download it to your machine, then you can go to download files, PDB format. Okay. You can also download the sequences, or you can design an API call to automate that process, and you can download thousands of sequences or structures a day that way. So that's the protein data bank. Its purpose is to house structures, and the structures in the protein data bank uh, were experimentally determined. Okay, they're not predicted structures; these are actual solved structures. They've been determined. Okay, there is another database called the AlphaFold database, and I don't have it pulled up, so let's go to it. So the AlphaFold protein structure database uh, is kind of the equivalent of the protein data bank, but it's predicted structures. So let's search for globin here. And you see that you pull up uh, different records to where, based just on the amino acid sequence, the structure was predicted, and these structures have actually been shown to be pretty accurate, very accurate actually. So, and there's many more structures available in the AlphaFold database than there are in the protein database. Why is this? Because it's very expensive in time and money to experimentally determine these structures. So the invent of AlphaFold has given us access to many more structures. Okay. Uh, there's also the Uniprot database. Now, the Uniprot database is kind of going to bring a lot of this information together into one spot. And you can search for globin over here. I have the alpha subunit of hemoglobin pulled up here. So it's a globin. And you can just scroll through its record. And you can download the record. You can download the structure. This has the structure in it. Uh, it also has the family and the domains. So sequence similarities. Let's see here. What all do we got here? You got the conserved domain database. You got Panther. You got ProSite. Several different databases here that you can link out to and go and look at their records. So these databases that I'm showing you are just a handful. Really, they're the databases that I like to use, but they're not the only databases out there. You also have the sequence of the protein here, or the protein domain. Um, you got different or different features of the protein domain. You got all kinds of information brought in here. It's basically, this database is for protein information, for one source of protein information. That's the Uniprot version of the name. Okay. We also have the protein database within the National Institutes of Health and the National Center for Biotechnology Information. And you can also search for the same protein. So we've searched for the same protein over here, and you get various different aspects of that protein. This also brings together information from a bunch of different sources about this particular protein. You can also find citation information sometimes in here. Now, I've used NCBI database many times, uh, but I got to tell you, it's not the most user friendly database out there. It's more in a form that looks more like a database structure here. It looks more like a machine readable structure. It's not quite as nice as the Uniprot database and how it looks. A little harder to link out sometimes, but you can definitely get the information you need from NCBI. So these are just a few databases that you can use to get information from for your studies on proteins and the protein sequences. Uh, I've told you a little bit about what you can do with some of those data. I've told you a little bit about what bioinformatics actually is and how it's really honestly it's a subdiscipline of data science, and it's also a subdiscipline of biology. So it's kind of a union between those two fields, and computer science as far as that's concerned. Uh, So you got to know how to do a whole bunch of different stuff, and you got to have a bunch of different knowledge to be able to be effective as a bioinformaticist or bioinformatician. Um, but you'll get there if you're just now starting your study of bioinformatics. Don't be overwhelmed. I mean, go out there, set a goal on something you're going to do, a study that you're going to do, and just do it. Work your way through it. Don't get overwhelmed when you don't know everything. Um, Seek out the knowledge that you need. Every one of these databases has help pages that can tell you more about the database. You can go to the about page on the scope database, and you can find all about it. But every one of these databases has it. Every one of these databases can be accessed via API from Python. So if you have to do a study and you need 20,000 sequences, uh, you can set up a script to pull those. You don't have to go in here and pull records one at a time. You can get any part of that record that you want. You don't have to download the entire record. You'll quickly find that your data gets huge. You end up with data files that are gigabytes, not just megabytes, but huge data files. And that's what a bioinformaticist does. It tames those large. They tame them large volumes of data and bring them down to something that can be presented and that can make sense and that answers a question. That's what a bioinformaticist or a bioinformatician does. So I hope this helps you out. I hope it gets you a little further down the line. If you like the video, please like it on YouTube and please check out my other videos. I hope you have a great day.